Okay, so I'm going to be talking about next generation mosquito control tools in Florida. And let's just jump right on in. So probably not a surprise to anybody, but that Florida is a hotspot for mosquitoes. And that's because, mosquitoes can't really maintain a constant core temperature. And so the tropical and subtropical climate here in Florida is, the heat, the humidity, it's perfect for them. And also due to the fact that Florida does have a lot of diverse aquatic habitats and mosquito eggs need water to hatch, it really allows all the diversity of the aquatic habitats allow so many species to find the right conditions for them. So because of our diversity in aquatic habitats, we do have a diversity in mosquito species. We're currently home to approximately 90 species. And of those 90 species, some of these are going to be nuisance spiders, so like the Mansonia we just talked about. And then also some that are disease-causing pathogen vectors. And just a super quick reminder, it's only going to be the female mosquitoes that are going to bite and potentially transmit pathogens. So due to the presence of the nuisance spiders and the pathogen vectors, Florida is one of the few states with extensive organized mosquito control programs. And so there's 66 state-approved programs currently, and there's definitely a lot of variation between those programs, mostly due to funding. And there's also going to be variation in the type of control methods that are used. Most are going to utilize an integrated mosquito management strategy. So using surveillance to decide when to treat, using multiple approaches to control the mosquitoes at multiple life stages, and also using public education as one of those strategies for control to try and get the public on board to help with control. And so you can see from this figure that I pulled from a study on this slide that in general, the most common method for controlling adults, which is in figure A, is going to be using a truck-mounted ULV spray. In figure B, you can see the different insecticides that are going to be used in the truck-mounted ULV trucks. And then figure C, you can see the different types of larval control strategies that are used by programs. So if most Florida mosquito control programs are using the integrated mosquito management, why should we explore additional control tools? Certainly new tools are going to aid in the fight against mosquito species that can be difficult to control with traditional methods. Additionally, resistance is becoming more and more common in certain species. And also additional tools can help fight in helping the fight against mosquitoes that can transmit pathogens that cause mosquitoes. And that's always beneficial. We're always trying to do that. So all the points I've just made about why we need new tools to help control mosquitoes are particularly true for two invasive species, Aedes aegypti and Aedes albopictus. First, I'll talk about Aedes aegypti. This is the yellow fever or dengue mosquito, its common names. They're both invasive species, and this species is widespread throughout the Florida Peninsula. Both of these mosquito species live in close association with humans, which means they like to take blood meals from us. And they also like to utilize containers that we might have around our homes and businesses for larval habitats, water-filled containers for larval habitats. The yellow fever mosquito, Aedes aegypti, is the primary vector in Florida and also worldwide of viruses that cause the diseases dengue, chikungunya, yellow fever, and Zika. And so out of those diseases, we have had outbreaks of dengue, chikungunya, and Zika in Florida within the past, I want to say 20 years. Yeah, within the past 20 years. So just talking a little bit about the Asian tiger mosquito, Aedes albopictus, again, just like Aedes aegypti, it's also an invasive species and it lives in close association with humans. This species can be found in rural locations as well as urban areas. It will feed on more mammals, but it will predominantly feed on humans in urban areas. And this species can also transmit all the viruses that I just talked about, but it's not the primary vector in our state. But places where Aedes aegypti in the world, or the yellow fever mosquito in the world, is not present, this mosquito has been present and has vectored these viruses that led to outbreaks. So when I'm talking about, container inhabiting, I wanted to just give you a better idea of what I'm talking about using this slide. And so here on this slide, you can see images of aquatic habitats where you can find the yellow fever and Asian tiger mosquitoes developing. Like I said, it's going to be the water-filled containers surrounding our homes and businesses. So, discarded trash. You may have heard about how mosquitoes can develop in a water-filled cap of water. It's true, I've seen it. Then of course you've got your plant pots and saucers, buckets, tires, bird baths, bromeliads. And so if you think about these containers and their openings and where they might be located around people's homes and businesses, it can be difficult to actually reach the mosquito larvae that are in the water of these containers with traditional larvicide methods. So that's one important reason why new tools can be beneficial for controlling them. And then additionally, I'm just going to give you some more information about one of the viruses that the yellow fever mosquito and Asian tiger mosquito can transmit, and this is dengue virus. It's the most important mosquito-borne virus affecting humans worldwide. And so there's approximately 50 to 400 million infections a year. It causes dengue and also severe dengue. 5% of the cases of dengue can develop into severe dengue. And you can see here on the slide the different symptoms of the two diseases. So dengue fever symptoms are going to be more mild, more like a flu-like infection. It's possible people may not even know that they're infected with dengue virus. And then, like I said, 5% of the cases can develop into severe dengue, and this is certainly going to have more serious symptoms such as shock, blood pressure dropping, severe bleeding, severe vomiting, and there is approximately 22,000 deaths a year worldwide due to severe dengue. And dengue is an ongoing challenge in Florida. We have had multiple outbreaks in the past 20 years. Just in 2020, there were 72 locally acquired cases in our state. To just the past few slides that I've talked about should have explained why they're needed. So now let's get into the next generation mosquito control tools. So recent advances in our understanding of the major mosquito pathogen interactions, combined with development of sophisticated techniques, have provided the opportunities to improve the classic types of control strategies that have been used in the past. So population suppression and population replacement. And many of these next generation tools are linked to either mosquito genome manipulation or mosquito associated microorganisms. And so we actually have had examples of next generation mosquito control being used, or tools being used in Florida. And that's what I'm going to talk about now, is those two examples. So first, I'm going to talk about the release of Wolbachia infected Asian tiger mosquitoes contaminated with paraproxyphene, which is a larvicide, in St. Augustine, and which was a collaboration between Anastasia Mosquito Control District and the company Mosquito Mate. And then next, I'll talk about the release of genetically modified yellow fever male mosquitoes in the Florida Keys, which was a collaboration, or ongoing collaboration, between the Florida the Florida Keys Mosquito Control District and the company Oxitec. So as I mentioned, the Asian tiger mosquitoes that were released in St. Augustine, they were infected with Wolbachia. And this is just a bacteria that can be, it's naturally found in certain mosquito species, and then it can be put into other mosquito species to have certain effects. And one of these effects that's possible in Asian tiger mosquitoes is cytoplasmic incompatibility. And what that is that in males that are infected with Wolbachia, their sperm is modified so that it can no longer successfully fertilize eggs. So if you look at this figure here on this slide, think about the blue male and female symbols being just normal wild mosquitoes, and they mate and end up producing healthy, normal male, female, and male mosquitoes. But if you, in the next line, wild female mates with a Wolbachia infected male, and that's actually an image of what the Wolbachia bacteria looks like. And in that case, you don't end up with additional mosquitoes. And so then what you can see is an overall reduction in mosquitoes over time. And so that's what that graph is showing there. So I think it's super neat about the strategy that was actually used by Mosquito Mate and Anastasia Mosquito Control District in St. Augustine, is that they didn't just stick with the ability of the Wolbachia to reduce the mosquito population. They even took it to a higher level. And I think that's just really neat. And so what I mean by that is that their Wolbachia infected males were dusted with paraproxyfen. And as I said earlier, paraproxyfen is it's a larvicide, and it's a really powerful juvenile hormone analog. And so it, can inhibit the development of immature mosquitoes. So in this figure on the slide, you can see that, these are males, a drawing of males, what it would look like, of males that have paraproxyfen on them. And so that's what the little molecules are on them. And they come in contact with, they can come in contact with larvae in containers that are around, just by going to visit themselves, or they can actually mate with females. And so that's what's being shown in the C portion of the image. And when they mate with the females, they can actually spread that larvicide paraproxyfen to the females. And so paraproxyfen is super potent in small doses. And so just those individual particles are potent enough so that even if it's the males themselves that have the paraproxyfen on them and they happen to go to containers where other mosquito larvae are, or if they've mated with females and then transferred that paraproxyfen to females, the females then go on to try and lay their eggs in other habitats where these larvae might be, it's going to kill the developing mosquitoes in those aquatic habitats. And so you have not just the oticidal males, so males that have the ability to, decrease the generation or decrease the population over time due to being infected with Wolbachia, but they're also self-dispersing or auto-disseminating this larvicide paraproxyfen. So it's multiple control tools in one. So I think that's so fascinating. So it's talking specifically about the trial that took place in St. Augustine. This took place in 2019, and future trials were planned, but they kind of got off course due to the pandemic. We all understand about that happening. So here you can see an image of the materials that were distributed to the public about the Asian tiger mosquito control program, to make the public aware of what was going on. And then also on this slide, you can see what the study site looked like, where the mosquitoes were released. And so it was just a small study site. So where the red dots are, that's where the mosquitoes were released. And then where the white dots are on this slide are cups that are black and then have water in them, and then some seed germination paper, so it's kind of like got a rough texture on it. And what happens is that these container inhabiting mosquitoes, they generally lay their eggs on the side of the containers. And so when you put that seed germination paper in, you're able to go to the site, pick it up, take it back to the lab and count the eggs. And so that's giving you an idea of population. And so that's actually how they calculated the impact of the release of these atom males in this site. And so what you can see in the bottom figure here on the slide, and this is a little bit backwards to what I was expecting, with the dark colors actually represent a lower percentage of survivorship rather than higher survivorship. So if you, in the first, in week 36, you can see where there was this was before the releases occurred, and then you can see that in the next few weeks, there's a decrease in immature survivorship. And so there is a reduction in the population, and then they're also reducing the number of immatures through that transfer of that larvicide. And so, like I said, this was just one trial that was conducted, and there were some additional plans for trials, and but they were kind of put on hold. And so hopefully there will be some additional trials with this planned in the future. But now we're going to switch gears and talk about genetically modified mosquitoes in the Florida Keys. So you've probably heard about this and, you know, wondered what exactly happened or what was going on. So in 2021, 2 pilot projects took place. Again, they were a collaboration between the Florida Keys Mosquito Control District and Oxitec. And Oxitec, just to give you an idea, is a biotech company, and they've been working on self-limiting mosquito technology for 20 years. And they've done multiple releases of these modified mosquitoes throughout the world. So this is not the first time that this type of technology was used. It was just the first time that it had been used here in Florida. So first things first, I'd like to talk about, you know, what a genetically modified mosquito is. And so it's just going to be a mosquito that has its genome altered to make it useful for reducing population size or controlling the spread of mosquito transmitted pathogens. In the case of the GM mosquitoes in the Florida Keys that were released in the Florida Keys, they were what's considered Oxitec's friendly strain of male yellow fever mosquitoes. And I think it's a silly name, but the reason they called it that is because the male mosquitoes don't bite. Like all male mosquitoes, they don't bite. And so I guess they were trying to stress to the public that they're the friendly ones because they don't bite. Not going to judge them from that name. So getting into what genetic modifications are actually made to these mosquitoes, there was just 2 genetic modifications. First, they added a color marker just that fluoresces under certain types of light that allows the researchers to be able to distinguish between the wild yellow fever mosquitoes and the genetically modified yellow mosquitoes. And so that gives you know, the researchers an opportunity to kind of understand what effect the release of these mosquitoes is actually having on the natural population. And so then the next gene is what's considered a self-limiting factor. And specifically in this case, this is one that induces female-specific lethality. And what that means is just the immature mosquitoes die before they can pupate and emerge as adults. And the way this happens is that it stops essential proteins that they're going to need for this, the next stages of their lives from being produced. So the males are going to have this gene, but they're not affected by it. And because the males are going to have this gene, they're also able to, when they mate with the wild females, they pass that gene on to the offspring. And so then the offspring, both the male and female offspring, have that gene, but since it's only a female specific lethality gene, only the female offspring are going to die. And so if you think about, offspring being like approximately 50-50 male and female, then you think about, 50% of mosquito offspring dying over time. This can lead to a reduction in mosquito numbers over time. So one of the questions, big questions that are asked about this study was how the mosquito eggs were generated to be used in the trials or in the pilot projects. So this image on the slide is just trying to give you an idea of how that is done. And it's this, as I mentioned earlier, so this company, actually, I don't think I mentioned it, but the company Oxitec, they are an English company, and that's where they have their headquarters. And so that's where they're going to be producing these mosquito eggs, and where they're going to be generating the mosquitoes. And when they're generating the numbers they need for all these for the trials they're conducting, they have tetracycline in the water. And so that tetracycline actually keeps that female specific lethality gene from being activated. And so everybody survives. And that allows them to get up enough numbers to be able to then have eggs to be used in future trials. But for any trials that are used, they're going to end up with adults that are not adults that lay eggs, and they're not going to be exposed to tetracycline. And so again, now that female lethality gene is going to be activated, and then those eggs are put in a capsule and sent to the Florida Keys, and then the eggs are just put in these boxes. And this is actually what is put in people's backyards. So it's just a capsule of the eggs, and water is added to the eggs, and then within the box, the mosquitoes, the male mosquitoes go through their development, and then the adult male mosquitoes emerge. And again, because there's no tetracycline, it's only going to be the male mosquitoes emerging that can then mate with wild female mosquitoes and lead to the reduction in the wild population. So as I said, there were 2 pilot studies that have taken place so far in 2021. Project A, Project B, no need to get things too complicated. And in Project A, they were just single point releases. And just as a reminder, these releases only took place in the Florida Keys. Aedes aegypti, or the yellow fever mosquito, as well as the Asian tiger mosquito, they don't fly very far. They're not strong flyers. And so they only fly approximately about 500 meters, just to kind of give you an idea that, these were small isolated trials. And honestly, these initial projects were to try and confirm that the things they were expecting to see, they did see. And I'll explain that better in just a minute. But so just in Project A, what separates Project A from Project B is that for Project A, there was just one box that had the eggs in it, and then the males emerged from it. And they wanted to see how their modified males, how far they flew in comparison to the wild males, and also how they were successful in terms of mating with the wild females. And they used monitoring tools to monitor both the wild population and the wild population, and then the with eggs and the adults. So that's what this trap here on the right side of the slide is used for. It's used for adult surveillance. So for the pilot Project A, as I mentioned earlier, in wild yellow fever mosquitoes, they generally only fly about 500 meters or so. And what they found was that the Oxitec males generally they had traps that were placed about 400 meters from the release location in Project A. And so they found that the males were able to fly that far, but they didn't see them generally beyond there. And so it showed that their ability to fly was comparable to the wild male's ability to fly. And this was shown for both Project A and Project B. Sorry, I should have explained what Project B was first. The only difference between Project A and Project B was that there were multiple boxes put out in one location for Project B versus Project A. That's the only difference. And also that project ran longer, and there were took place in more areas. So that's the only difference between them, just more boxes and a longer trial. So I just wanted to make sure I brought that up just super quickly. So like I said, the flight range was found to be the same for the Oxitec males versus the wild males for both Project A and Project B. And then additionally, they found that there was effective mating. So they looked at different doses, which would just be a different number of eggs put in the box. And so they found that, so they're trying to work out like how many eggs should they put in each box. And so that's what this table is showing, is them, trying out different doses to see, what's that right number to put in each box. And so they found that there was effective mating with their high doses. And then of course there were some sites that just didn't have enough mosquitoes for there to be an effect on the numbers. But in those sites where there was enough mosquitoes, they did find that there was an effect of the male mosquitoes basically serving as having that larvicidal effect, because that gene is being transferred to the female progeny, and then those female, well, actually it's transferred to both, but the female progeny died as larvae. So that's what that image is showing, is fluorescing larvae that are dead, which is a good thing for mosquitoes. So as I said, the only thing with Project B was that there was multiple sites or multiple boxes at a site. And then they also wanted to see, is this technology allowing the, are the females that are mating with the males, are they being successful in reaching some of those habitats where it might be difficult for traditional larvicides to reach, like what I had talked about earlier. And so they did find that, yes, there were the Oxitec progeny found in, what are considered cryptic locations. And so this is evidence that, yes, this technology can be used to control larvae in these areas that the traditional larvicides may not be able to reach, because the mosquitoes are much better at finding themselves than we are. So that was what took place in the 2021 trials. The EPA did approve Oxitec to be able to conduct more trials, the Florida Keys Mosquito Control District and Oxitec to conduct more trials in 2022. And so this image on this slide is just to give you an idea of what they have planned for the trials in 2022. So they want to continue with trial B and do some more small neighborhood studies and looking at the proportion of the population that is treated. So really getting into more of looking at the effect of this population suppression tool. So actually measuring it to see, is it starting to have an effect on suppressing the population? And so that, and you can just kind of see that, there's, they're trying to find out if different configurations of the boxes might work in a better way. So that's kind of what's going on between, the differences between the different trials, B and D. And then also for E, they actually want to look more into that ability of the males, their flight, how far they can fly. And so they're actually going to be doing that through actually marking them with, well, actually they're already marked. So doing a better job of looking into that dispersal. And then also this time looking into their longevity and how that compares to the wild males. So definitely this project, so this is, I actually took this from a screenshot of this from a webinar that Oxitec did, I think last month or the month before. So this trial has actually, what they're doing now is community outreach. So FDACS has approved it. Everything's moving forward for 2022. And like I said, they're out in the field, they're meeting with community residents and talking to people about if they're willing to host a box in their yard of the that contains the eggs. And they're starting to do the pre-release monitoring. And then they're going to start Project B up first and then conduct Projects D and E later. I have no idea what happened to Project C. I don't know. So that just kind of gives you an idea of what's going on this year and what'll be happening later in the year. And I do know that in addition to Florida, Oxitec is actually going to be starting trials in California with this release of the genetically modified yellow fever male mosquitoes. So for those of us who are lucky enough to be in the locations where this next generation control technology is being used to reduce these Aedes container inhabiting mosquitoes, I always like to come back and talk about what can you do to reduce the mosquitoes around your home. So as I've talked about, these are mosquitoes that the Asian tiger mosquito, the yellow fever mosquito, they're not going to be the ones that you find in your canals, your lagoons. You're not going to find them there. You're going to find those in the containers around homes and businesses that end up getting filled with water. So, it's a great idea to try and dump out these containers once a week. For the containers that you can't dump, such as like bird baths, you can put mosquito dunks or bits. Those items can be found at your local hardware and just general purpose stores, and that contains the bacteria BTI. And that's a very safe bacteria. It's very specific. It's only going to have lethal effects on mosquito larvae and midge larvae. So it's only going to kill midge larvae and mosquito larvae. It's super safe. It can be even put in drinking water. And it's not going to have any effect on any other type of wildlife that might be drinking that water. And then if you happen to have bromeliads around your house, you can also put the mosquito bits in the bromeliads, or you could use, a stream from a water hose to rinse them out every week. Or additionally, one thing you might consider is contacting your local mosquito control program. Some programs will actually make site visits and see if they can find the source of where the mosquitoes are coming from in your yard, and potentially provide you with mosquito fish that can be used for biological control. So that's really what I was going to cover. And I certainly want to thank Julian for the invitation to give this presentation, and I'd be happy to answer any questions.
Thank you very much, Eva. We do have time, and I see some questions in the chat. Would you want me to read the questions, or you're okay reading the questions? How would you want to handle that? You're muted.
So sorry, I was muted for a moment. So I am uncomfortable reading them. So let's see.
We had some questions about the Oxitec mosquitoes.
Yes,
first.
absolutely. So Frank, no, it's not going to be every year. It's really just going to kind of be like a year by year basis. And honestly, it's based on EPA approval, FDAC's approval. So we know that there are going to be trials in 2022 for any trials that take place in 2023. If they want to have trials, then of course they're going to have to go through all of, get all that approval. Let's see. So is the color change passed down to the offspring? Absolutely. Okay, so Frank asked, is there data to say that the mosquitoes in the test area were reduced? Such a good question. Really, at this point, the goal was, they were kind of, as I showed in one of the slides, they were kind of like working out, what dose or number of eggs to put in the different boxes. And so they were trying to figure out what is going to be needed to start seeing that population reduction. So, and honestly, just making sure that there was no female larvae, that, there was mating that was taking place between the male and the females. So to my knowledge, there wasn't a reduction seen in these initial pilot projects, but it's hoped that that will be seen in the future trials. Okay, I'm gonna move on to the next question. So the function of, what is the function of the TAV gene in the GMO mosquitoes? I'll be honest, I forget what the the name of these genes are. So what I can tell you is just one gene that's modified is going to be the one that makes the Oxitec mosquitoes fluoresce. And then the other one, I think actually that's the TAV gene, that's the female lethality gene. So that gets passed on when males mate with females, that gets passed on to their offspring, and then any female offsprings die before they emerge or before they pupate. Hope that was a good explanation. Okay, so Rochelle asked, during this summer, some of our fields are flooded, which increases the mosquito populations. What would I suggest before going out in the fields? Fantastic question. So I recommend either, so for me, out in the summer, I'm obviously a pale kid. I wear the long sleeve light material to help with not getting sunburned. So if that's something that you wear and you feel comfortable in, wearing long sleeve pants and shirts, that can reduce, the amount of skin that you have available to mosquitoes. And of course, you can also wear repellent. There's certain active ingredients that the CDC recommends, as well as the EPA. So if you want to use a repellent that has been approved for use by those two groups, then you're going to want to look for one that's containing an active ingredient of either DEET, oil of lemon eucalyptus, Picardin. Oh gosh, I'm like blanking at the moment. But if you go to the CDC's website, they have a list of all the active ingredients that they recommend being used in the repellents. So Carol asked, what has the public response been regarding the release of the Oxitec mosquitoes?
And actually, I think that's a great question, and I wanted to ask the same. And what I wanted to add was also the mosquito control professionals and how that interaction has been conventional insecticides versus some of those, because we know that typically, at least the public would not be extremely supportive of conventional insecticides, but there might be some reluctance also adopting some of the new generations. So I guess it's a little bit pick your poison for the public. So great question.
Yes, I also think so. So I actually have, I had some temporary employees that worked in my lab last winter and spring, and then they went on to work at Oxitec. And then I have a PhD student that works full-time at the Florida Keys Mosquito Control District. And so certainly, this is something I've been interested in, and I've asked them, and they have, personal experience. They're out in those neighborhoods. They're actually, going, talking to people, asking them if they can place the boxes in their yards. And what I hear every time I ask this question is that the local public is actually super supportive. They have done a lot of education to get the message out. And so when the people actually, whether it's Florida Keys Mosquito Control District or Oxitec, when the employees go in their yard or, go to the front door and ask them, they most of the time already know about it, and they're just super supportive. They're excited about it. And that has actually been something that has actually been unexpected, but a very good thing that there really hasn't been, a local pushback. They're actually really excited. And so I think that, certainly there are people that are against this technology, but I wouldn't bet the people, the local people in the Key West, or sorry, in the Florida Keys, they're very supportive.
Thank you. I do not see any additional questions in the chat. Anybody with question in the live audience?
Eva, great presentation. I learned a lot today. I had a very general question. We don't have any yellow fever in Florida, correct?
No, no.
But we do have dengue and we do have Zika transmission sometimes.
Sometimes. I love that you added that at the end. Thank you. That's very helpful. So no, we do not have yellow fever virus transmitted here in Florida. And luckily there is actually an effective vaccine against yellow fever. So that's a really fantastic, really, a wonderful technology that can be used for that disease. And in terms of chikungunya, dengue, and Zika, those are considered exotic viruses. And so they are not going to be constantly transmitted in our natural population of, there's not going to be transmission occurring all the time. What's going to need to happen for transmission to occur here in Florida is someone from a location where it is endemic. If we talk specifically about dengue, someone gets infected with dengue, they come here and are infected. One of our local yellow fever mosquitoes bites that person, gets infected, and then the virus infection spreads throughout the mosquito, makes it to the salivary glands, and then the next time that mosquito bites someone, that virus can be transmitted to somebody else. So it's going to take extra, when you think about like transmission that's taking place all the time versus, you know, it having to be brought in every single time for an outbreak to occur. It's just, it's going to be more difficult. And so we really are lucky in that these viruses aren't endemic. However, we do have some endemic viruses, and those are going to be the ones like West Nile and Eastern equine encephalitis virus, which, you know, I can cover another time.
Thank you. Thank you.